Good afternoon, one and all. So the next topic in the chapter gene abnormality is polymerase chain reaction. Okay, so though it is given under this section, that is gene abnormality, it is not a it is not a gene abnormality, but it is a process of DNA amplification. Okay, so which is mainly used in many biotechnological process. Okay, so wherever we want to study the sequences in the DNA, okay, we should have enough copies of the DNA. Okay, so this technique will help in getting the n number of copies or multiple copies of the target DNA. Okay, so it is useful technique in many biotechnological work. Okay, so PCR is a laboratory technique for generating the large quantities of a specified DNA. Okay, so as I said, it is a laboratory technique which by which you will be able to get multiple copies of a specified DNA. Okay, so it is like a Xerox machine in an office. Okay, so in an office, like a Xerox machine is used to take the multiple copies of the document, isn't it? Okay, so which is very essential in not only in our routine life, in many places we need to have multiple copies of the documents. Okay, similarly in biotechnology, polymerase chain reaction is a technique to produce multiple copies of the DNA. Okay, how much important is Xerox machine in an office? Similarly, polymerase chain reaction. Okay, so this instrument is very essential on all the biotechnological work. So it is an amplification technique for synthesizing multiple identical copies of any DNA of interest. Okay, so we should understand that. So what is polymerase chain reaction? It is a DNA amplification technique. Okay, so by which you will be able to produce multiple copies of our target DNA or the DNA of interest. Whatever the DNA which we want to study, we can produce the multiple copies of that DNA with the help of polymerase chain reaction. So that is why it is called as DNA amplification. Amplification means like increasing the number, amplifying. Okay, so here we are with if you have a single piece of DNA, with that you can, by using polymerase chain reaction, we can produce multiple copies of that. Okay, so producing technique to produce multiple copies of a DNA of interest, that is called as polymerase chain reaction or DNA amplification technique. Okay, so coming to the principle of PCR. Okay, so PCR, in this technique we have a double-stranded DNA of interest, so which has to be denatured. Okay, so first step is the denaturation of double-stranded DNA. Okay, so what is this denaturation? Denaturation is nothing but separation of the two strands of DNA into two individual strands. Okay, so DNA, we know it is a double helical structure. Okay, so if you want to produce the copies of the DNA, you need to separate it. Okay, so that separation technique, okay, separation of the double-stranded DNA into single-stranded DNA is called as denaturation technique. Okay. Next, after that, after the denaturation, so now the DNA strands are separated. Okay, so the second step is a renaturation, or it is also called as annealing. Okay, so annealing or renaturation. So in this process, what you do is the separated strands of the DNA is allowed to hybridize with the primer. Okay, so primer means it is a small strand of nucleotides, most probably an RNA. Okay, so this small strand of RNA is hybridized with the separated strand of DNA. Okay, so because we need our intention by this PCR is to produce the multiple copies of the DNA. Okay, so we cannot produce the DNA without the help of a primer. Okay, so it is a initiating strand of the nucleotide. Okay, so to which the polymerase enzyme can add the nucleotides. Okay, so if there is no primer, polymerase chain reaction cannot synthesize the new strand. Okay, so there should be a small initial strand to which it will, the polymerase enzyme will add the nucleotides. Okay, so that is very, very essential here. Okay, so RNA primer has to be added here. The second step is the joining of primer, that complementary nucleotides of the primer with the flanking DNA of our that is the DNA of our interest. Okay, so it will get base paired. Okay, so at one end of the DNA. Okay, so that technique is called as a renaturation process. So it is a in simple way we can say it is a hybridization, a complex of primer and the template DNA is formed. Okay, so this PCR is more or less same as that of our DNA replication process. Okay, just imagine the DNA replication process. So it also works in the same fashion. Okay, so primer template duplex is formed now. So this will be used for the DNA synthesis. Okay, so who is the enzyme participating here is the DNA dependent DNA polymerase enzyme, because it is a replication, similar process is similar to replication here. Okay, DNA copies are synthesized from the DNA itself. Okay, so the enzyme polymerase enzyme is DNA dependent DNA polymerase enzyme. Okay, for as transcription and all, you remember that is DNA dependent RNA polymerase, reverse transcription is RNA dependent DNA polymerase. Okay, so here we are synthesizing DNA from a DNA, so the enzyme is DNA dependent DNA polymerase enzyme. Okay, so then this polymerase enzyme should have some specifications also that I will tell you. Okay, so these are the three steps involved in the main three steps in the process of PCR. Okay, that is first step is denaturation, that is separation of the two template strands of the DNA. Next is the renaturation, that is hybridization of the primer with the template strand. And the last one is synthesis of the DNA, that is polymerase enzyme will add the nucleotides to the primer. Okay, so and helps in the growth of the DNA. Okay, so that is called as that is the steps and the principle involved in the polymerase chain reaction. Next, coming to the procedure of the PCR technique. So here, before going into that, we will see what are the requirements of the PCR. Okay, so the first one is the, okay, so this requirement and the procedure will be discussing together in this. Okay, so the first requirement is the template DNA. Okay, so that is our DNA of interest. Okay, so it should have a minimum length of 100 to 35,000 base pair length. Okay, that is a length required for the template strand of the DNA. Okay, then we need the two primers. Okay, so we should have the primers, that is which is called as, which is synthetic oligonucleotide of length of 17 to 35 base pairs. Okay, so at least up to 35 number of base paired nucleic acid. Okay, so is required. Okay, so it should be complementary to the flanking target DNA. Okay, so target flanking DNA means the DNA which contains the promoter region. Okay, so promoter region in that we have studied. Okay, so it is a place where the primers will bind to it. Okay, so where more enhancers and activators are also involved in this. Okay, so it is a place, it is a recognition place where the primer binds to the DNA, that is called as flanking DNA. So this is not actually participating in the transcription. This portion will not get transcribed. Okay, so so identifying the flanking region of the DNA and the primer will binds to that DNA. Okay, so that is the, so the we need enough concentration of the primer. Okay, so concentration of the primer is also very, very important. So otherwise DNA itself, two DNA strand itself will get base paired. Okay, so to avoid that, high concentration of the primer has to be maintained here. Okay, so that the primer will hybridize with the DNA. Otherwise what happens is the two strands of the DNA will get hybridized complementary. Okay, so yeah, to avoid that, high concentration the primer should be provided here. Then the our deoxyribonucleic acid. Okay, so the here ATP, then that is deoxy adenine triphosphate, deoxycytosine triphosphate, similarly DGTP, DTTP, all the four different types of purine and pyrimidine nucleotides. Okay, so that is enough concentration of these purine and pyrimidine nucleotides has to be provided. And the last one is polymerase enzyme. Okay, so here the polymerase enzyme which we use should have capacity to withstand high temperature. It should not, this enzyme should not get degraded with high temperature. Okay, that is, it should be thermostable, because in the denaturation process, that is first step in the PCR, where we separate the both the strands of the DNA. Okay, so at this stage of the PCR, we need to use high temperature of 90 to 95 degrees centigrade. Okay, so when we expose the DNA strand to this temperature, the enzyme, which is, PCR is an automated instrument. Okay, so all the requirements will be put together and it is like it works automatically. Okay, so you are not adding the primer after the denaturation process, nothing like that. So all the requirements will be put together and the instrument will run like that. Okay, so automatically. Okay, so this primer also will get exposed to this temperature. Okay, so primer should have the capacity to withstand this temperature. It should be thermostable. So most commonly used is the Taq DNA polymerase. Okay, so not primer, the polymerase should have the capacity to withstand this temperature. Okay, so the polymerase used here is the Taq DNA polymerase, which is obtained from the bacterium Thermus aquaticus. Okay, so this is the source of Taq DNA polymerase, or TAQ DNA polymerase. So it should have the they are thermostable polymerase enzyme. Okay, so that is the reason they are used for this reaction. Okay, so I mistakenly I told primer, so the polymerase enzyme. Okay, so it should be thermostable. Okay, so it should be able to withstand the high temperature which is used during the denaturation process. Next is coming to the actual steps involved in that. Okay, so here it involves repeated cycles of amplification the target DNA. Okay, so first step involved here is a denaturation process. Okay, so denaturation means here this is the process of separation of the two strands of DNA. Okay, so both the strands of the template DNA is getting separated. Okay, so how it is separated? That is by raising the temperature to about 95 degrees Celsius. Okay, so for about 1 minute. Okay, so when the temperature is increased, what happens is two strands of the DNA get separated. Okay, so it becomes two individual strands. Okay, second step is the renaturation process or annealing. Okay, so renaturation or annealing means here this is the hybridization of primer with the template DNA. Okay, so that process is called as renaturation. So two strands are separated by denaturation. Then after that, joining of the primer with the template DNA strand, which is called as renaturation. Okay, since there is hybridization or joining, it is also called as annealing technique. Okay, so this annealing technique is achieved by decreasing the temperature or slowly cooling the setup temperature to about 55 degrees centigrade. Okay, so at this temperature, primer will base pair with the complementary region of the target DNA strand. Is that clear? Okay, so the primer will binds with the DNA strand at the flanking region. Okay, so flanking region I told already. Okay, so it is the region where the recognition sites are present, where the primer will at this place the primer will have the complementary nucleotides. Okay, so it will base pair with the complementary nucleotides in the DNA strand. Okay, so this process is called as renaturation or annealing. Okay, so high concentration the primer ensures annealing between the each DNA strand and the primer rather than two strands of the DNA. Okay, so that is what I've told in the starting. Okay, so primer concentration is very important here. It should be enough concentration of the primer in order to ensure the binding of primer with the DNA. Okay. Then after that, the third step is the, so now denaturation over and renaturation over. The third step in the PCR is the synthesis technique. Okay, so synthesis technique here, this is the synthesis of DNA. Okay, so that is the nucleotides, that is ATP, GTP, CTP and TTP is added to the PCR, no? So that will get the polymerase enzyme, DNA polymerase enzyme will mediate the binding of these nucleotides with the primer. Okay, so at the 3' end of the primer. Okay, so this primer, as per the nucleotide sequences in the template DNA, the primer will mediate the transfer of the complementary nucleotides to the polymerase enzyme will mediate the transfer of nucleotides to the primer. Okay, so the nucleotide which is carried by the polymerase will be complementary to the nucleotide sequence of the nitrogenase bases in the template DNA. Okay. So thus the primer get extended. Okay, so by joining the bases complementary to the DNA strand. Okay, the synthetic process in the PCR is quite comparable to the DNA replication process in the leading strand. So however, the temperature has to be kept optimal as required by the enzyme DNA polymerase. Okay, so at this temperature, at this condition, the temperature should be optimum to the DNA polymerase enzyme, so that DNA polymerase can work effectively to bring the nucleotides to the growing strand of the DNA. Okay, so for Taq DNA polymerase enzyme, the temperature required is 75 degrees Celsius. Okay, so and for E. coli DNA polymerase, it is around 37 degrees. So based on the polymerase enzyme used at this stage of the PCR, temperature has to be controlled. Okay, so it is an automated instrument. Okay, so it itself will manage the temperature. Okay, so it itself will adjust the temperature. Okay, so during the denaturation to 95, during the renaturation to 55, and during the synthesis phase to 75, or synthesis phase temperature is based on the polymerase. Okay, so you have to set the temperature, so it itself will control the temperature. So reaction can be stopped so whenever, if the enough multiplication, if enough copies of the DNA is the size, then we can stop the technique by raising the temperature to about 95 degrees centigrade. Okay, so how many cycles of the PCR has to be repeated? So based on that, we need to set the machine. Okay, so automatically it will get stopped once the required cycles of the PCR gets over. Okay, so each cycle of the PCR, it takes about 3 to 5 minutes. Okay, so in the normal practice, PCR is carried out in an automated machine. Okay, so once the required cycles gets over, it gets automatically stopped. Okay, so then you will have, so in each cycle the number gets doubled. Okay, so 2 to 4, 4 to 8, 8 to 16, 16 to 32, like that. Okay, so as a result, so what is the required number of the DNA? Based on that, you can set the cycles of the PCR also. Okay, you can see here in this, so this is the template strand of the DNA. So here these are the nucleotides, and the red colored are the these are the primer. Okay, so in the first stage, that is denaturation process, temperature is raised to 94 to 96 degrees centigrade. So the two strands get separated. This process is called as the denaturation process, separation of the initial strand of the DNA. Okay, once it is separated, second step is the annealing. Okay, so that is like 50 to 60 degrees centigrade. Okay, so you can see annealing or renaturation means hybridization of the primer with the DNA. Okay, so you can see here the primer is attached to the template separated two template strands of the DNA. Third step is the addition of nucleotides by the polymerase enzyme. Okay, so you can see here this primer is getting extended like this with the addition of the nucleotide. So this primer is getting extended like this. Okay, so this is how the extension of the strands of the DNA takes place. Okay, so here you can see in this in the after the first cycle, this is one cycle of the PCR. Okay, so from two strands of the DNA, four strands of the DNA is produced. This four will get separated, one, two. So this is the template strand here, and this is the template strand here, and this is the template strand here, and this is the template strand here. So these are the primer added to hybridization of the primer. So here this is the next denaturation. Okay, so after denaturation, next step is the renaturation. Primer adds to the or gets hybridized to the template strand. Okay, so the new strand of the DNA is produced. Okay, so here these arrowed ones are the new strands of the DNA. Okay, so in the second step, 8 strands of the DNA is produced. Okay, then again in the third step, this will get denatured, then renatured with the hybrid primer, and then the synthesis, addition of the nucleotides to the primer by the polymerase enzyme. Then in the third stage, there will be number of 16 number of DNAs. Okay, so like that, the polymerase chain reaction will continue. Okay, you can see here this is the original strand, new strand. After the one cycle, 2 copies. Second cycle, 4 copies. After third cycle, 8 copies. After fourth cycle, 60 number of copies. Okay, this is how the DNA will get doubled. Okay, so that kind of DNA amplification technique is called as the polymerase chain reaction. Okay, so that is about the PCR or the polymerase chain reaction, which is an amplification technique of the DNA. Okay, so from a single strand of the DNA, you will get multiple copies of the DNA. Okay. Next, we are coming to the DNA sequencing technique. Okay, so this is also, though it is this topic is also given under this chapter only, but it is not an abnormality. It is the sequencing of the DNA. So you are going to identify what is the sequence of nitrogenous bases in the DNA. So understanding the sequence of nucleotides in the DNA, that technique is called as the DNA sequencing technique, or it is also called as DNA mapping technique. Okay, so identifying the number of nucleotide sequences. Okay, so the A, T, G, C, like that. What is the order of nitrogenous bases in the DNA? Okay, so that is called as DNA sequencing technique. So this is very, very important. It is a fundamental requirement in the biotechnology. Okay, so when you want to study about any genes, or if you want to do any kind of genetic engineering process, that is like recombinant DNA technology or any kind of gene therapies. Okay, so or if you want to produce a particular protein. Okay, so what is the sequences of nitrogenous base? What is the sequences of nucleotides in the DNA? Okay, that should be known to us. Then only we can do any kind of manipulations in that. Or if you want to produce a therapeutic DNA, okay, so you should know what should be the correct nucleotide sequences in a gene. Okay, so understanding that sequences in the DNA is very, very important for the biotechnology for the biotechnological work. Okay. So and also for the cloning, for gene manipulation. Okay, so for all these things, accurate nucleotide sequences should be known to us. Okay, so the one technique which was developed by Maxim and Gilbert, the two scientists, in the year 1977. So this is called as the DNA sequencing technique, which is also called as Maxim Gilbert method. Okay, the Maxim Gilbert technique of DNA sequencing. Okay, so here what they have done is, so nowadays there are more sophisticated techniques to understand the sequences of DNA. So many assays are also available to understand the sequences and to study the nucleotide sequences. So this was one of the initial technique, and which is very easy to explain also, and we will go into the technique. So here the first what they do is, one strand of the source DNA. Okay, so you have a target DNA. So one of the strand of the target DNA, if one strand sequence is determined, means you can say the other strand sequence, because other strand will be obviously complementary to that. Okay, so we can write the sequences in the other strand. Okay, so first one strand will be separated from the DNA, and that will be radio labeled. Okay, so one of the source DNA will be labeled at one end with the phosphorus 32. Okay, so this is a radioactive isotope. Okay, so this one strand at one end will be radio labeled. Okay, so that radio labeling is just to understand where if this strand is one side is radio labeled, and then from that end we are going to see the nucleotide sequences. Otherwise sometimes we will have the, if one end is not identified, then we will have a confusion whether after identifying the nucleotide sequence it is from this end to that end or that end to this end. Okay, so to avoid that, one end will be radio labeled. Okay, then labeled DNA will be distributed into 4 samples. Okay, so yeah, you take 4 set of tubes, and all these radio labeled DNAs will be placed, equal quantities of the radio labeled DNAs will be placed in the 4 separate tubes. Okay, so one strand is taken, one of the strand is radio labeled, and these strands are separately placed in the 4 tubes. Okay, so equal volume of this DNA strands are placed in the separate 4 tubes, and each sample is on all these 4 tubes, 1 2 3 4 tubes now. Okay, so these 4 tubes are subjected to treatment with a chemical which will specifically destroys one of the nucleotide. Okay, so one of the nucleotide or 2 nucleotides. Okay, so that is C, G, then A plus G and T plus C. Okay, I will tell you that. Okay, so thus the DNA strands are partially digested at 4 samples at site G, A plus G, T plus C and C. Okay, so as I said, you are taking 4 set of tubes. Okay, 4 set of tubes containing radio labeled DNA. Each tubes you are going to treat with the chemical which can digest the DNA at particular nucleotide. Okay, so first tube you are treating with a chemical which will digest the DNA or breaks the DNA at G. Okay, so wherever G, guanine nucleotides come, so the DNA can digest that chemical can digest the DNA at the place where G is present. Okay, and the second tube will be treated with another chemical which will digest the DNA at places where A and G is present. Is that clear? Okay, so at places where A and G is present. Okay, so wherever A and G comes, at that place this DNA will be digested, or it will breaks the DNA at that place. Digestion means it is breaking the DNA at that place. Okay, so wherever A and G is there. Okay, and in the third tube DNA is treated with another chemical which will breaks the DNA wherever T and C is present. Okay, so it will breaks the DNA at places where T is present as well as C is present. Okay, and the fourth tube you are adding a chemical which will digest the DNA at places where cytosine is present. Okay, so where C is present. Okay, so these are the 4 chemicals which you are using in these 4 tubes. Okay, so 4 tubes taken with radio labeled DNA which is treated with the chemicals which will digest the nucleotide at particular place. So first tube will digest wherever G is present. Second tube the chemical will digest or breaks the DNA at places where A and G is present. Third tube will digest or breaks the DNA wherever T and C is present, and the fourth tube will breaks the DNA wherever C is present. Okay. Okay, so this results in the formation of a series of labeled fragments of varying length. Okay, so this will when they breaks the DNA, so you will get many fractions of the DNA. Okay, so in each tube there will be different fractions of the DNA. Okay, so actual length of the fragment depends on the site at which the base is destroyed. Okay, so if G is present at the second position, okay, so it breaks at second position. One more G is present at the 9th position, it will breaks at 9th position. Like that, that means that second strand of the DNA will have 8 number of nucleotides. First strand of DNA will have 1 number of nucleotide. Okay, so like that. So based on the place where the particular nucleotide is present, based on that, the length of the fragmented DNA will vary. Okay. So thus, for example, if there are C residues at position 4, 7 and 10 nucleotides away from the radio-labeled end, then the treatment of the DNA that specifically destroys C. So for last tube will destroy C, no? Say third tube and last tube. Okay, so we'll destroy the DNA at wherever the C is present. Okay, so there you will get, if the C is located at the 4th position, 7th position and 10th position. So it is breaking at 4th position, and it will break its other DNA at 7th position. It will break some other DNA at 10th position. Okay, so you will get a fragment of the DNA with 3 nucleotides, 6 nucleotides and 9 nucleotides. Understood? Okay, so it is breaking at 4th position. So that means that that strand of DNA has 3 number of nucleotides. 4th position it is broken. So from the radio labeled end, 3 number of nucleotides. Okay, so 7th position it is broken. That means from the radio labeled end, 6 number of nucleotides. At 10th position it is broken. If it breaks at 10th position, then from the radio labeled end, the DNA fragment has 9 number of nucleotides. Okay, like that. Don't think that in all where these are the different number of different fragments produced. Okay, so why in the 9 why the DNA fragment with the 9 nucleotides, why it is not broken at 4th and 7th? It is not like that. Okay, so it is like wherever that particular nucleotide is present, there it breaks. Okay, so you will get many fractions, many fragments. Okay, so some fragments will have in some fragment it is broken at 4th position, in some fragment it is broken at 7th position, in some fragment it is broken at 10th position. Okay, so you will have some fragments with the 3 nucleotides, some fragments with 6 nucleotides and some fragments with 9 nucleotides, like that. Okay, okay, so then labeled DNA fragments obtained in the 4 tubes are subjected to electrophoresis side by side, and they are detected by autoradiograph. Okay, so then all these fragments of the DNA will be separated from the tubes, and that will be subjected to electrophoresis. Electrophoresis, you know, you have studied, you remember we have studied about rocket electrophoresis and different types of electrophoresis technique in the in microbiology last year. Okay, so there what we do is, with the application of electric current, the fragments are separated. Okay, so here the fragments will get separated based on the size. Okay, so more the fragment with the less size will move to a longer distance. Fragments with more size or the larger size will move only to a shorter distance. Okay, so it is a method of separation. Okay, then the sequence of the bases in the DNA can be constructed from the bands on the electrophoresis. So after the separation, it forms a bands. Okay, so from the bands you will be able to identify the nucleotides in the particular position. Okay, so you see here, you will understand better. So this is the DNA double-stranded DNA or the sample DNA. So here we are separating the single strand of the DNA, and in the second step, that will be radio labeled at one end. You can see a small blue color spot here. So from this is the radio labeled end. Okay, so from this radio labeled end, and this DNAs will be taken in the 4 different tubes. Okay, so the first tube, second tube, third tube and 4th tube. Okay, so and this will be treated, each tubes will be treated with the chemicals. First tube will be treated with a chemical which will digest the G nucleotides. Second tube will be treated with a chemical which will digest A and G nucleotides. Third tube will be digested with a nucleotide which will digest T and C nucleotides, and 4th tube will be treated with a chemical which will digest the nucleotide C. Okay, so here then after that, after this, okay, so once the DNAs are fragmented with the chemicals, then it will be subjected to electrophoresis, and after the separation, it will be read. The plate will be read under autoradiography. Okay, so why the autoradiography is used? Because here these fragments of of DNAs are radio labeled. Okay, so these radio labeled compounds can be wherever it is separated based on the size. It will get separated. Once it is separated, where it is separated, okay, so that can be easily identified by, because it is radio labeled. Compounds are, DNAs is radio labeled. Okay, so you can identify the presence of the radio labeled DNA by autoradiography. Okay. Okay, so after that, once this is separated, you can see here. So this here in this tube, the fragments will be formed like this. Okay, so this tube is digesting the DNA at wherever G is present. Okay, so you see here, this is the place where G is present. Okay, so G is at the 4th position. Okay, so you will get a nucleotide of three nucleotide, DNA strand of 3 nucleotides. Understood? Okay, so you see at this position. Okay, so there is a strand. Okay, so that means this strand has 3 nucleotides. Okay, so and then where the next G is coming at this position. Okay, so this is 1 2 3 4 5 6 7 8 9 10th position. Okay, so you will get a strand at the 9th position, because at 10th position D it is breaking. Okay, then this is the, sorry. Okay, then this is the original strand of the DNA. Okay, so this is the original strand of the DNA having 12 number of nucleotides. Okay, so these are the 3 fragments of the DNA which you are getting from this tube, from the first tube. Okay, so in the second tube you are treating the DNA fragment with a chemical which will digest A and G. Okay, so you see here in the A, yeah, okay, so even though it breaks at this position, since there is no strands of the since there is no nucleotide beyond here, so you will not be able to identify this digestion actually. So whereas in the 5th position A is present. Okay, so you can see here G is already there in the 4th position. Okay, so you will get one strand in the at this 3 position, because it will digest at G also. So this will be 3 number of nucleotides in this strand of DNA, and one more digestion at this 5th position. So you will get a strand with 4 nucleotides. Can you see here? Okay, so this indicates that here also fragment is there, here also fragment is there. That means this is G, because in this tube, from this tube, at this position you are getting a fragment, at this position also you are getting a fragment. Okay, so that means this is G, whereas here also one band is there, but that band is not here. That means it is A. Understood? Only in this tube you are getting this fragment. Okay, so whereas that fragment is not there in this tube, that means that is only A. If here in both the place, if you are getting the fragment, that means that is G, because in this, from this tube as well as from this tube. Okay, similarly for in this tube, you see this will digest the at T and C position, and this will digest at C position. Okay, so here you see the second one is T. Okay, so you will get a fragment with a single nucleotide, that is A alone. Okay, and third is also T. Okay, there also you are getting. Okay, so a fragment with the 2 nucleotides A and T. 1 and 2 is here now. So with that you can identify that. Okay, then here it is G and A, that will come in these tubes only. So here you see there is C. Okay, so at 6th position. Okay, so you can see here at 5th position you will get a fragment C. Okay, so that same fragment you will get from here also, because this will digest at T and C. This will digest only at C. Okay, so whereas wherever the T fragmentation is happening, there is no band in this tube. Okay, so if you are getting band in this and in this tube, that indicates that that is C. If you are getting band in this and in this, that indicates that is G. If here and here same fragment, then it is G. If here and here same fragment, then it is C. If here alone, then it is T. If here alone, then it is A. Okay, so like that we can identify the all the, you see this picture, very observe this very picture clearly, considering this fragment of the DNA, so you will understand it. Okay, so this is how you can detect the sequences of nitrogenous bases in the DNA. Okay, so if one of the strand is identified, nucleotide sequence is identified, then the other strand is complementary to that. Okay, so by which you can understand the number of or the sequences of nucleotides in the DNA. Okay, so that is about the Maxim and Gilbert method of sequencing of nuclear DNA. Okay, so that is all for today. Thank you so much. Thank you. Time is also getting exhausted. Okay, so thank you all of you. Thank you. I'll stop here.